Today's presentation is What's New in Physiologically Based Pharmacokinetics in the Simcyp Simulator. Our speakers are Dr. Mark Lovern, Dr. Masoud Jameh, Dr. Jean Din, Dr. Udo Amaka Azuruike, and Dr. Felix Stadter. Mark is currently a Senior Vice President of Commercial and Product Strategy at Certara's Simcyp division. Masoud is also a Senior Vice President in our Simcyp R&D division. Jean is a Senior Research Scientist at Certara Simcyp. Amaka is also a Senior Research Scientist at Certara Simcyp. And finally, we have Felix, who is a Senior Research Scientist at Certara's Simcyp division. Welcome to the presentation, everybody. I'll now turn it over to Mark to begin.
Well, thank you, Suzanne. It's a pleasure to speak with everyone today, and I'm really excited to be joined by such an august panel for this presentation. And I'm going to lead off with a few introductory slides around what's new in PBPK and Simcyp itself. Also, I'm going to start off with just a brief introduction of what is physiologically based PK and how is it often used, because some on the phone might, or in the meeting may not be familiar. So, one of the key distinguishing characteristics between physiologically based PK and classical PK, which has been around for quite some time, classical PK models tend to be focused on predicting what systemic exposure is to a drug, so the exposure in the bloodstream. PBPK, and there are other compartments often in classical PK, but they don't necessarily have a direct physiological interpretation in terms of an organ or tissue group. And that is the case with PBPK. Is just about every compartment you have represents an organ or tissue. And then the processes in with regard to distribution and elimination are as closely modeled after physiologic processes as feasible. So, for instance, many of the flows are mediated by the blood flow, which makes complete sense. One of the things that PBPK can be used for is to predict scenarios that would not be feasible using classical PK. And so here you see illustrated a sort of a generic PBPK model. The blue boxes are typical applications of the approach. So we look at one of the major ones is looking at drug-drug interactions. We can also look at food effects, virtual bioequivalence. We can explore racial differences, and then also special populations such as patients with organ impairment or pediatric or geriatric patients. And we can also, again, and also populations with reduced cardiac output. So a few words about PBPK in the literature. If you look at the plot on the left-hand side, this is data from PubMed, and it's showing the publications per year since 1981, I believe it is. Actually, the first occurrence was 1987, now that I remember it. It was essentially, it's showing the volume of publications in that year in which that turned up as search results for PBPK. So you can see that clearly over time, this methodology or this subject continues to grow exponentially. On average, the growth rate in publications is about 5% per year. And last year, there were 440 publications which had that search term in them. Alongside this, track record of increasing volume of publications, there's this very nice quote by Ananda et al. From a publication just last March. It says, currently the use of PBPK models to replace human evaluation for predicting drug-drug interactions is widespread in the pharmaceutical industry and accepted by worldwide regulatory agencies. So, as I mentioned earlier, drug-drug interaction is one of the major applications of PBPK. We'll talk about some others as we go through the presentation today. So, that's PBPK in the literature and media. Let's now take a look at the regulatory perspective. specifically, I'm going to lead off with a notable quote that's on the FDA website. And here we have, instead of a drug-drug interaction example, here we have a virtual bioequivalence. And essentially, the key message here is for a topical gel of 1% diclofenac sodium, essentially they were able to approve a generic formulation without the benefit of a human bioequivalence trial. In addition, several guidances were released in 2022 by both the FDA and the ICH, all of which at some point in the guidance advocate the application of PBPK approaches. So the two from the FDA are on pediatric dose selection and also food effects. From the ICH, we also have a pediatric guidance, but also have a drug-drug interaction guidance. And all of these specifically call out PBPK as a worthwhile approach. So we've talked about PBPK in general. Now let's talk about Simcyp Simulator. This is Certara's solution for performing physiologically based PK models, and it is the world's most sophisticated PBPK platform. We have extensive libraries on demographics, developmental physiology, and as well as ontogeny of drug elimination pathways. We have an unmatched body of science. We have 10 advanced mechanistic organs, 29 subpopulations, and over 115 compound files, which we'll talk about what those allow us to do here in just a little bit when Jean presents. We can, one of the major applications is predicting in vivo pharmacokinetic exposure based on in vitro data. And most of the top 40 biopharmaceutical companies are consortium members, including all the top 10. In addition, 11 regulatory bodies, including the US FDA, are consortium affiliates. So here are some, as I mentioned, major areas of application or impact. One being is that we, as just mentioned a few moments ago about the virtual bioequivalence, our technology is often used to get clinical trial waivers and also extrapolate to special populations, including pregnant women, pediatrics, geriatrics, and special disease populations. And in instances where you cannot fully waive the trial, the simulator can be used to greatly reduce sample size, to streamline the overall size of the trial, which, as most people understand, will reduce both the cost and the timelines associated with it. So one other thing we're very proud of here in Simcyp is that our technology has been used to make over 300 label claims and counting to the benefit of 90 drug development programs. And this table here summarizes these programs by therapeutic area, and this list continues to grow every year. All right, so that's what's new with at a high level with regard to Simcyp and PBPK modeling. And now we're going to spend a little bit of time talking about some of the new features in the Simcyp simulator with version 22, which we just released. Excuse me. And it should be noted that we have many more new features than we can talk about in this 1-hour presentation, but we tried to select some of the ones that we think are going to have the biggest benefit or impact for our users. So first up today is Jean Din, who joins us from Seattle, and Jean is going to talk to us about the addition of new and upgraded compound files. So welcome, Jean. We look forward to hearing what you have to say.
Thank you, Mark. I'm happy to talk to you all today about the new and upgraded compound files that we are introducing in V22 for Simcyp. So if you could advance to the next slide, please.
Of course.
Thank you. Just to give you all an idea of the structure of Simcyp, you can think about there being three main buckets of data within Simcyp, and these are the data that we kind of collate together to come up with these predictions. The first set of data is the systems data. You can think about it as like the populations and the data that we put in to get an idea of populations of your interest, and then the variability within that population. And my colleague Amaka is going to talk more about that. The second piece of information is the trial design right there in the middle. This is how the study is being done. This is the study that you want to simulate. And then lastly is the drug data or the compounds data. And this is where my team focused their efforts on this year. And really this is for the development of the compounds going into the simulator, and the information that we pull together to make this happen are all the physiochemical properties of the drugs, the metabolism, the absorption pathways, solubility, etc. We put that all together to come up with our predictions of drug PK, and in some cases, the pharmacodynamics as well in your population of interest. So if you can move to the next slide, and we'll discuss the compounds that we worked on this year and where you can find them. So this slide gives you an overview of what our team did this year. In blue are the library compounds. And so these will be available to you in the simulator. And what how a library compound differs from a research compound, which is highlighted in purple, was that there was enough data to not only develop the compound, but also verify the compound. The research compounds, at the moment, there was not sufficient data to do the verification. Nonetheless, these are available to you on the Simcyp members area in case these are valuable to you in your simulations and your research. In green are compounds that were actually developed in V21. However, these use the GI anatomy that was originally built into Simcyp, but the GI anatomy, the original GI anatomy was used in earlier versions. Now we've upgraded to the new GI anatomy. Now these compounds can be introduced into the simulator because previously these used the new anatomy. So that's great. And in gray, there's only one compound there, but this was something that we looked into because this was a consortium member request. However, unfortunately, there was not enough data to develop this, but it's on our radar, but we did look into this. You can see the types of compounds that we worked on. They number CYP substrates. You can see those are the most numerous compounds that we worked on. There's one UGT substrate, which is morphine. There's also 4 transporter compounds and actually 1 large molecule, and actually one's an ADC. So that's really good. And that's actually a research file. So you'll see that on the Simcyp members area. In addition to developing new compounds, we've also updated current compound files as well, and those are listed there below. And these upgrades could be the addition of new induction parameters, addition of new metabolites, or the addition of the pharmacodynamic model to the existing compound file. And finally, we also worked on adding CYP1A1 into the simulator. Previously, we relied on CYP1A2, so now this has been added in to provide more physiological relevance to the system.
Gene, can I ask a quick question here?
Absolutely, Mark.
This is quite a large list of new compounds. Are these all cherry-picked by the consortium members, or are there other criteria that get them on the list for this year?
That's a great question. So primarily, I would say that the consortium member votes on this because they probably had a need for it in their own research, but sometimes research purposes on our end motivate us to add a compound, or sometimes too, if we see a compound that's being recommended by regulatory bodies to be used as either a probe substrate or as a perpetrator, that also gives a higher priority to be added to this list.
Okay, great. So are we ready to move on?
Yeah,
Great.
let's do that. Okay, so I'm just going to walk you through each of the compounds. And as you saw on the previous slide, the majority of substrates that we developed fall into that CYP category. And of these, many of them hit the CYP3A4 pathway, either as a CYP3A4 substrate or a perpetrator. And then in the case of 3 substrates here, both. So you can see vitratinib, cobistatin, and voriconazole are both a CYP3A4 substrate and perpetrator. The only exception to this would be duloxetine, which was the only substrate where it did not touch the 3A4, surprisingly, but it does hit CYP2D6 and 1A2. So yeah, those are the CYP compounds there, and the other CYP pathways are listed if necessary. If you go to the next slide, we can then go over the transporters. So as I mentioned before, we developed 4 transporters for V22. As you can see, these compounds are certainly far more complex. While they were developed to study transporter activity and drug-drug interactions, you can see there's a significant CYP component for each of these as well. And in the case of dronedarone, it's also being developed as a PGP inhibitor. Right?
Does it, when you have transporters involved, is there, I'm guessing it's a more complex, there's a lot more complexity to actually developing the compound file, right? Probably a lot more data that has to go in and verification and all that.
Yes, and more complex models too, as well, because you have to think about the organ where the transporter is expressed and active. Absolutely. And so here's just a summary slide of the remaining additional compounds and updates. As I was saying previously, morphine is our one lone UGT substrate, but it is there. And as I was saying before as well, ritonavir and favirenz, this data is quite interesting. There was some data from one of our colleagues in industries suggesting that these 2 compounds, there's a CYP2C8 induction. So we've added information there so that you can add it if needed. As I was saying before, there's 1 large molecule, but the ADC drug conjugate is also available as well. And then lastly, the addition of the pharmacodynamic models to existing models. So again, not only can you anticipate PK, but potential drug effects as well. All right. Next slide. And then lastly, I do want to highlight the support documentation that is available for these compounds. Now, these are all going to be available to you at the members area, but importantly, each of these documents details the clinical studies that were used to verify the PK of these compounds. You can see how these were tested, and so these are also important for regulatory submission, but also if you want to know a little bit more about how we did it, that's available for you at Simcyp as well. And right now, 100% of those compounds are V21 summaries are available there for you. We are working on V22 as it was just recently released, but we are hoping to have these on the members area as well.
Great, thank you.
Thank you. And I have to highlight my lovely team. So just a round of applause to them for their hard work this year. Greatly appreciate it.
Thanks, Jean. Quick question for you now that, I mean, you've done all these compound files. What are you working on now? More compound files, or do you have another project that you're
I'm working more compounds files. Compounds.
All right. Thank you so much.
Thank you.
All right. Now I'd like to invite Amaka to come on screen and talk to us about the expansion of our population library.
Hi, Mark. Good afternoon. Can you hear me?
Yes, absolutely. You're coming through loud and clear.
Okay. All right. I guess I'm just going to talk about the additional populations that we've added to the Simcyp library for version 22. If you just advance the slide for me, please. Okay, so Jean has already talked about this, about the structure we have in Simcyp by separating systems data from drug data. So for this presentation, I'm just going to focus on the systems data. So if you just make one click. So for the systems data, what we really look at is the population library. And in the population library, what we're considering is implementing physiological parameters that will properly describe the population of interest. So the overall aim is to develop a virtual population that is best representative of the population you're interested in. So for this, we collect certain data, so like the age distribution of the population, height and weight, information like tissue volumes, tissue composition, tissue blood flow, which is actually dependent on the cardiac output you mentioned, so whether it increases or decreases, and then plasma proteins, which determines the level of binding of the drug or the compound of interest. And then we also put in information about the expression of various enzymes and transporters, which can vary between one population and the other, and also putting information about the kidney function. So the renal function can be very instrumental for compounds that are mainly cleared by the kidney. So what we did for version 22 is that we added 5 new populations to the Simcyp library. These are the populations that are highlighted in red. So we added a geriatric gynaece population, and then we added 4 distinct North American ethnic populations, and I'll explain why we did that in a minute. So that brings the total number of populations we have in Simcyp to 29, which you will agree with me is quite a number, and we're always increasing this number every day. But also, in addition to these 5 populations that were developed newly, we also did an extensive review of the existing obese and morbidly obese disease populations. So kind of like incorporating more recent physiological data.
Sorry, I'm trying to advance. There we go. Okay.
Okay, so I guess the key thing is, so why did we have to develop 4 North American populations? And I think this quotes by the FDA commissioner really just highlights and brings to the fore why this is important. And so he mentions here in this quote that the US population has become increasingly diverse. And it's really important that for clinical trials clinical trials should better represent the diversity of the population. And actually, the truth about the matter is that historically, some clinical trials already incorporate this diversity, but we were not accounting for these in our simulations. And as you can see from this chart at the top, you can see that in certain parts of the US, you can have a specific ethnic population really accounting for a significant number. So for instance, the African American in certain areas can account for up to 40% of the population. So to better represent or replicate these clinical trials that are currently being considered, we have developed 4 different North American populations. So a North American white population, a North American African American, North American Asian, and a North American Hispanic. Now within Simcyp, we already have this multiple populations feature. So what this allows is that you can then use this feature to simulate up to 4 different populations at the same time within one clinical study.
Oh, can you choose the mix? Like, can you decide, like, when you put these 4 populations, like what percentage of each you can put in there?
Yes, definitely. So, so this one is showing you, I've just used an example of 4 populations. So if you look at this clinical trial, it said you had 12 Asians, 12 Blacks, and 12 Caucasians. So if I was going to replicate this study, I would select the multiple populations, but only select 3 population, I'll only highlight 3 populations. And then I can specify the number for each population. So saying that the Caucasians are 12, and then the Blacks are 12, and the Asians are 12. So as much as the clinical trial you're trying to replicate, you can replicate it exactly. So what you're simulating is as much as possible the clinical trial you're trying to represent.
Can I ask one other question here? So we have other Asian populations that were already existing, but they're at other geographies and things. So we expect that there would be differences between like a North American Asian and someone that's actually in Asia. I mean, is that diet or what is it? It's probably, what would be causing that difference, do you think?
Actually, that's a good question because whereas when you see, so we have within Simcyp, we have a Japanese Asian population and a Chinese population, and we also have a South Korea population in the members area. So, but when you come to North American population, the North American population is not one purebred Asian, so it's a mix of the different populations. Although based on the US census, the greatest population are those of Chinese heritage. But then you also have an influence of diet. So within Simcyp, when we build a population, we consider both intrinsic and extrinsic factors. So by the time you put in all those elements, your predicted PK may be slightly different from the Asian community you're considering in the US because of its mixed diversity compared to the pure Asian as we have with either the Japanese or the Chinese population.
Okay, thank you. Thanks. Very, very helpful. Interesting.
Okay, so I think for this slide, I just wanted to highlight why it's really important for us to account for these different ethnicities. So historically, we've accounted for different ethnicities in Simcyp by having the Chinese and the Japanese. But more importantly for the North American is that when you have a simulation with a mixed North American population, the exposure of the drug might not be the same in that subpopulation. So this is an example with pravastatin. So pravastatin is a transporter substrate. It's a substrate of multiple transporters. So MRP2, OATP1B1, 1B3, and OAT3. So, and if you look at the exposure of pravastatin, you can see that the exposure of pravastatin is higher in white American compared to the African American, which means that the African American population are clearing prevastatin faster. And so the question is why? Because if you were not thinking about those differences and you were just taking the mean average exposure for the whole population, you wouldn't realize that certain subsets of that population are driving the PK in different directions. So the key reason why you're seeing this change between the exposure in the African American and the Caucasian white American population is because of the frequency of OATP1B1. So within the African American subpopulation, you have a greater frequency of ultra rapid transporters. So most of the population are either extensive transporters for OATP1B1 or ultra rapid transporters. So that means that the uptake of pravastatin into the liver in that subpopulation is much quicker than you would have for the Caucasian population, thereby resulting in about 30% decrease in the exposure compared to the white American population. So that was just kind of like highlighting why we needed to develop multiple populations for the North American. So one other thing that we did for this project was also to add to the simulator a Chinese geriatric population. I know, Mark, you already mentioned in the beginning why it's difficult for us to account for changes between the young and the elderly. So this really highlights it because we had a general Chinese population and also a Chinese healthy volunteer population, but we also needed to extend that general population to 95 years because this subset of the population has a high degree of polypharmacy. And we know that in addition to the polypharmacy, the clearance of certain drugs in elderly population is decreased relative to the young population. So we developed this Chinese geriatric population to be able to capture these differences in PK as shown with these three different drugs between the young population, the young Chinese population, and the elderly Chinese population. So that was really the background for adding.
I mean, that's a 50%. That's almost about a 50% increase in the AAC.
Yes.
That's pretty significant.
Yes,
Yeah.
it is. Yes. And then finally, and I mentioned that we also had an extensive review of the existing obese and morbidly obese population. So we've had this population in the simulator for quite some time, actually. But if anyone who is following the news about obesity, you're very aware that the prevalence of obesity has increased significantly. But that also translates to the demographics of the population that are considered obese. So what it means is that if we need to update the population to better reflect the reality if we're using these virtual populations. So what we did in the first instance is to extend the population data based on more recent data from the Health Survey of England. And then we've also incorporated changes to enzymes and transporters that we did not previously consider in the previous obese population. So as you can see on these graphs on the right, you can see that for certain drugs, the activity of the enzymes are changed in the obese population relative to the control, which will then translate to a change in the PK of that drug or for drugs that are metabolized by those enzymes in the obese population relative to the control. One other important thing that we implemented in this version was to better account for the changes we see in blood flow to different organs in obese population. So in a normal population, we have an absolute blood flow to different tissues. But with the obese population, the increase in fat size translates to an increase in cardiac output. But what this means is that for certain organs, that increase in cardiac output does not necessarily translate to the increase in absolute blood flow. So what we have done in version 22 is to implement a dynamic tissue blood flow so that enables you to have a relative change in blood flow to different organs depending on the change in BMI. So we're currently doing an extensive verification of compounds for this population, and that will be available shortly.
So,
next. Go on.
Sure, sorry. Well, I was just wondering, you said some organs receive differential increases in the blood flow as with BMI. What are, I mean, obviously adipose tissue probably is one of those, but what are some others that like disproportionately the blood flow changes in this population?
Okay, so I if you think about it, for blood flow in the obese populations, it's kind of like divided into, the organs can be categorized into three classes. So for some organs, the blood flow increases with increasing cardiac output. So like the adipose, for instance. For some other organs, regardless of the increase in cardiac output, the absolute blood flow is the same. So for instance, like the brain, the absolute blood flow is the same. So what it means is that because in Simcyp, we account for blood flow as a percentage of cardiac output. So the percentage of cardiac output will now decrease with increasing cardiac output. It will not remain the same. Whereas for other organs like the heart, the percentage of blood flow to the heart increases with cardiac output. So we're able to account for these dynamic changes with change in BMI, which we did not have previously in the simulator.
Okay, thanks for explaining. Thank you, that's very helpful.
Okay. And then just kind of like to highlight what Jean said previously, we also try to prepare population summaries for these different populations that highlight, first of all, the physiological parameters that have been implemented in building these populations, but also contains some performance verification of this population against certain Simcyp compounds. So currently in the simulator, we have population summaries for all the populations that are listed here on this screen, and most of them are in version 21. And there's a team that is currently working to extend these population summaries to version 22 and version 23 concurrently.
Great.
So I just want to highlight on this slide everyone who contributed to the work that was done for the populations project in this version.
Great, thank you, Amaka. That's very, very interesting.
Thank you, Mark.
All right, so now I'd like Felix to come on and tell us about sub-Q administrations and what we've done to enhance our capabilities there. So welcome, Felix.
Thank you, Mark. So this project was split into two parts. The first part is for therapeutic proteins, with which we will start. And the second part is for small molecule drugs that we'll do towards the end of this presentation part. So for therapeutic proteins, the question is, why is subcutaneous dosing so important? And if you look at the drug approvals of the past years, then we see that 1/3 of approved antibodies are for subcutaneous dosing. And this is likely to increase because the patient compliance is improved and the treatment costs are reduced. But the big challenge is, what is the bioavailability of any novel antibody, of any novel protein that you develop? And PBPK modeling can help in answering these questions, can help in supporting you for a first dose after subcutaneous dosing. When we looked into the literature, we saw that there's no PBPK model that can predict the bioavailability a priori. And there are certain difficulties there. The first difficulty is, how do we define the subcutaneous injection site? It's not a distinct organ such as liver and kidney. The second issue is the clearance pathway of our proteins are often unknown and cannot be incorporated, and the information by in vitro assays is missing.
So Felix, does that mean you can have clearance of the compound right in the site of administration, in addition to like through the systemic clearance, which is probably more what we're used to in the small molecule context?
Yes, so there are proteases directly at the SC site that can cleave proteins and peptides, and there are also antigen presenting cells to which I come in 2 slides time. So on the next slide, we see how we handle the definition of a subcutaneous injection site. So what we have done is to use labeled albumin studies where they measure the travel distance from injection until the albumin gets deeper into the tissue. And this is what we've used to define the SC site. What we've incorporated are 4 different sites of the body: arm, abdomen, back, and thigh, to allow you to predict the bioavailability differences for the different sites. And on the right-hand side, you can see a prediction for IgG, where we see the differences in peak concentration and time to peak concentration between the different sites. On the next click, you can see that those differences are in line with what is published in the literature. So this is from a review of over 50 different antibodies, where we can see the peak concentration that is 14% higher in thigh versus abdomen and 8% lower in arm versus abdomen. And we're predicting these numbers with the Simcyp simulator with our included database for the different sites. What we've also included as a fourth point is deconvolution of clinical data that is also incorporated into Simcyp. On the next slide, what we see there is the inclusion of antigen presenting cells as one of the clone's pathways before reaching the systemic circulation. So what happens to an antibody is that after the subcutaneous injection, it distributes into the lymphatic system. And the task of the lymphatic system is to filter the fluid for all foreign substances. And the filtration is done by antigen-presenting cells, by macrophages. So what they do is to have a high uptake rate into the cell, but they also have a protective mechanism because we don't want to get rid of our endogenous immunoglobulins. So there is a protective pathway called the FcRn receptor that protects the antibodies from degradation. But initially, we have a very high concentration after injection, and this saturates transiently this protective pathway, meaning we have a higher clearance and meaning we have a reduced bioavailability. And this is what we predicted with the Simcyp simulator and what we've incorporated into the current subcutaneous absorption model.
So it's a dynamic sort of process then, the interaction here with the antibody.
Yeah, it's a dynamic process. So onto the right-hand side, what you can see here is the free fraction of this receptor. So for the black line, a typical organ, you don't see any changes. But the blue line are those antigen-presenting cells directly at the SC site, and you can see initially a drop in this free fraction, and that's the saturation, and then it returns back to the baseline level.
Interesting.
But what is the model performance? So what we try to do is to use IgG for our model performance because all antibodies are based on IgG. So by using the changes in binding affinity to this receptor or to the uptake rate, we can predict and also the bioavailability for an antibody. So what we see here is plasma concentration from individuals on the left-hand side after subcutaneous dosing of IgG. And we see that the absorption rate, peak concentration, time to peak concentration, and also the elimination rate is predicted in accordance to the observed data. In the middle, what we see is also IgG data that are where the samples have been taken directly from the injection site. So what we're seeing here is the removal of IgG after subcutaneous dosing. And that gives us confidence in our volume and in our lymph flow that we're using in our physiological database. And lastly, what we also see is typical PK parameters. So we have a time to peak concentration of 4 to 5 days and a bioavailability of around 60 to 70%, which is in line with what we see in the literature. So to conclude, for therapeutic proteins, we can predict bioavailability a priori by incorporating those pre-systemic clearance pathways and by knowing our drug, our binding affinity to the receptor, and our uptake rate. Now we're moving on to small molecule drugs. So for the first time, we introduced a subcutaneous dosing for small molecules as a well-stirred compartment. So we have the SC site as a well-stirred compartment available for the minimal and full PBPK model for substrates and all inhibitor positions. The only parameter we need from the drug site is the partition coefficient that is predicted via Rogers and Rowland as for all the other compartments that we're using. We incorporated the same for injection site that I told you before for therapeutic proteins, and we also incorporated deconvolution of clinically observed data. And that was the end of my presentation. I'd like to thank the entire team that put their great effort in implementing the subcutaneous absorption model in V22.
Thank you, Felix. Appreciate it. Great presentation, great functionality. And now we're going to switch over to Masood Jamie, our head of our science group, that's going to tell us about simultaneous fitting. And then we're going to chat a little bit about some of the features we didn't get to talk about today in detail, and also what will be potentially coming in version 23. So over to you, Masood.
Thank you very much, Mark. And if you go to the next slide, I can just, yes. I thought before we go to this project, possibly many people, they may not be aware that in the Simcyp simulator, we can do fitting. So you can use it as a bottom-up tool, but at the same time, you can use it as a top-down. There are various fitting algorithms that are incorporated in the simulator. You can do individual fittings. Many methods are incorporated, and you can do population fitting as well. So we have incorporated the QRPM method as well as MPAC. So parametric and non-parametric maximum likelihood methods are incorporated. And if people are interested, they can look at the paper where we compare the performance of model versus the standard Bayesian samplers that it just published last year. For this year, we came up with some challenges that people they have many different clinical studies, and they wanted to use those clinical studies to inform the PBPK model. In this case, both Jean and Amaka, they mentioned about the development of the compounds or populations. In this specific case, if you are looking at, for example, cimetidine, and we want to find out what is the Ki value for cimetidine that is performing and it can predict it. We can use one clinical studies, two clinical studies, and use that that clinical data to inform the model. And obviously, you will be able to predict that specific model well. In this case, we use midazolam, the first line in the table, and the predictions versus observed value, or observed versus predicted value, they were good. But then when you are going to apply that model to other cases, it may or may not be as good. So with the simultaneous fitting module, we can fit several of these clinical studies at the same time. Up to 10 clinical studies, they can be saved at the same time. And obviously, when you go to population fitting, the covariates, the number of subjects will be different, covariates will be different, the model structure will be different, and all of these can be handled in the Simcyp simulator now. So we can use it for determining some specific compound, key compound values. Alternatively, we can apply it for determining some population parameters. In the next slide, we have shown a very common case that if you want to predict drug PK in a special population like pediatrics, then you have to know what is the ontogeny of 3A4. There are several different publications that they have come up with several profiles for the 3A4. In our case, we have used midazolam through different studies. We have done very sophisticated back calculation to be able to come up with a specific ontogeny. But with the new capabilities, we can combine different drugs in different clinical study and come up with a more robust ontogeny for 3A4, and we can do it for any other key parameters when we are developing a specific population. Maybe it becomes more exciting if you go to the next slide, and it shows you that for pravastatin that Amaka just showed you, we know that polymorphism is a major factor. And this polymorphism, it changes from one population to another one. So in the first step, we have used the polymorphism in Caucasian data to inform the drug model. When we have established the drug model and we are happy with that one, then we can apply that one to, for example, Japanese, where we know that the phenotype and the function of transporter, they are different. And because they are different, now we can use different clinical studies where pravastatin is being given to identify the phenotype differences between in the Japanese population. So it is a combination of developing a compound and then applying that for informing the specific population. So this one is give you lots of power to be able to inform either your compound model or your population model and develop more robust PBPK models. One more thing that we have done in version 22 for fitting, in previous version, we were able to model and fit 4 different species at the same time. So we have animal simulators, rat, mouse, monkey, and dog. And in version 22, we now have the capability of fitting all the models at the same time. So if you have a specific parameters, obviously they should be sensible parameters like parameter specific intrinsic drug parameter across the board is the same, and you don't know it, you haven't measured it, or you don't have confidence in that, you can fit it across the board for all the different species. One more thing that we have done as part of our population fitting, as well as individual fitting, we are now can handle the below level of quantification, and the standard and most robust models, which are the M5, M3, and M4, they have been incorporated in the simulator. Again, people who are in the PBPK area, they are very familiar with this one, but if anybody interested, maybe we can provide more information later on. And that's what it's for this project. Obviously, all of the work, it was done by the project team, and I would like to thank all of them for the great job that they did.
Yeah, that seems like there's a lot of flexibility in terms of how, what kind of data you can fit and the algorithms and things. Are there any visions for further extending that capability in any future release, or we feel like we've got something, what we need for now?
Obviously, always there is the model can be improved. One thing that we are using and we are working on right now is we want to get to the Bayesian framework, and this is something that we are working. As you know, in the PBPK analysis, you can inform your parameters and the whole of the fitting algorithm if you have a distribution or prior distribution of the parameters. This is something that we are working on in this area. Yeah.
Okay, so now let's maybe mention a few things, additional features in version 22 that we were not able to talk about in detail today because we didn't have a lot of time. So Masood, tell us some of the other stuff that
yes, there are many more stuff. As you said, we didn't have time to go through the one. One of the major expansion was we have a mechanistic kidney model that has been used very frequently. We expanded that. Now the transported in the kidney, that they can be applied through IVIVE, and using absolute abundances, the number of transporters have been expanded. And we can model precipitation in kidney, that for toxicity can be important. The physiological parameters that they have been updated because we continuously look at the data and update those. Another thing that we looked at, the mechanism-based inhibition is one of the type of drug interaction. We expanded this capability and introduced new models. We did an overall review of the prediction, and hopefully soon we will be publishing that one. One more thing that we did on the absorption, we keep adding more and more advanced feature to our absorption model. We had an excipient model, but it was using the inhibitor place. Now we have developed a standalone excipient model. We mainly cyclodextrin as well as others. So we have provided a library for that. And one more important thing about this excipient model is that it's capable of modeling the displacement if you have more than one excipient in the model. And you can model the permeability effect as well. So it's not only solubility or dissolution, it can model the effect of excipient on permeability. We further develop our dermal model, both in vitro site and in vivo site, new sites. So you can apply the drug in many more number of sites simultaneously or one after another. New type of skin they were being introduced. One new module that we introduced is long-acting injectable. This is a new module for us. Route of administration, it is a common route of administration that people are using that one. We continue to expand our virtual bioequivalent module. Now the rectal as well as intravaginal can be incorporated in VBE. The dermal module can be incorporated, and specifically looking at, for example, metabolite. You can look at bioequivalence from the metabolite side. So more expansion on that side has happened. I think two more things. Obviously, the pediatric pregnancy and lactation population, they expanded, and many features they were added specifically to the pediatric for people who are working on ADCs, antibody drug conjugate. Now we can use the pediatric module to simulate those. One exciting new product that we have released with version 22 is the Simcyp Designer. For many years, people, they wanted to be able to manipulate the model in Simcyp, main Simcyp simulator, and now they will be able to do that using the Simcyp Designer.
Okay, so a lot is in the version 22. I'm curious, how big of a team supports all of that? And like how many roughly, and I assume there's a lot of testing that has to be done. Give me just a feeling for every year, like the amount of effort that goes into all of that.
It is massive. It is massive amount, and sometimes people outside they may not appreciate it. But just give you some numbers. We have about 50 or more number of scientists working on the simulator. Something around 30 software developers are working on the simulator, and we have 5 or 6 dedicated testers that they are working on various softwares that we are developing. So thousands of hours of work on the research side, on the development side, and on the tester side and testing side. It goes in to be able to provide a very robust and worthy product for the clients. And
Yeah,
we
I can imagine. Yeah, go ahead. Sorry.
No, I just briefly for version 23, lots of more exciting products are coming through. As you know, the new developments, all our new, most of our new developments are guided by the consortium. So they are deciding what we do next. We were very pleased to see that the confidence in the absorption area and the application are expanding, and consortium member, they asked us to expand further those areas. We are going to model add the PBPK modeling for endogenous biomarkers. Something has an impact on the clinical application as well as regulatory side. Database on the compound, Jean mentioned that. She and her team, they are continuously working on that side. pH mediated drug-drug interaction, this is another area of, I would say, concern or regulatory requirement that this model can help. One thing that we are doing, again, mechanistic, we are implementing the mechanistic model for drug therapeutics and small molecule drug interaction, as well as new modalities, oligonucleotide, bows, ASOs, and siRNA, that this new to PBPK model, they will be added to the version 23. So many more capabilities are going to be added to the simulator.
Wow, that's exciting stuff there. I think now we should take a moment and see if there are some questions from the audience. So, if the other panel members will come on camera in case this question is best addressed by one of you. So one says, first question, are the compound summaries available to everyone or only consortium members?
Hi, they are available to everyone with a Simcyp license. Just log into your Simcyp, into the Simcyp members area, and you have access to it.
Right, but it's not available to the general public. You have to.
That's correct.
You have to pay to play, so to speak. We gotcha. Okay, this one's a little open. How can we predict bioavailability from the new subcutaneous model? Is that? I mean, it's a little open-ended, I think. There. Anybody want to offer some thoughts on that?
Yeah, so what we do in the simulator is to take all the pre-systemic clones pathways and look what the eliminated amount is to calculate bioavailability. So what we need is to have the information about the drug. But the easiest way is what I showed you with those antigen presenting cells. You can just switch them on the drug absorption screen in the new model, and it will be automatically incorporated, predicting the higher catabolism in macrophages if there's the transient saturation, so that can be accounted for. If proteases play a role directly at the SC site, I already mentioned that there's a big gap, so we don't know really the abundance of those proteases, and we don't have those in vitro assays to inform the model. So this is a bit where there needs to be more work in the future.
Great, thanks, Felix. So here's another question. How do you optimize the probability the FDA will accept simulations of metabolism DDI in place of clinical DDI studies? What validation of the model is necessary to get FDA acceptance? Maybe that's one for you, Masood. What do you think?
Yeah, it's a very good question, obviously, and this is the very first step. I think anybody is going to submit a PBPK model related. So the regulatory guidance, they ask you to show that your model is capable of doing that. And the compound summaries that Jean, as well as Amaka mentioned for compound summaries, population summaries, they are showing the capability of the model that we have. Obviously, the degree of confidence differs from one area to another area, and we are addressing those. We had a very recent a video that we released it, and we looked at the number of the cases that we have been successful to look at 3A4, for example, and the number of submissions that inform the drug labels, and how many of those cases we have managed to save a clinical study. It was your slide that you show, Mark. More than 90% of the cases that we had a case that 3A4 was a drug, the metabolizing enzyme, we have managed to at least save one clinical study. When we are talking about the saving clinical study, we shouldn't trivialize that. There are lots of hard work and good data are required to be able to do this type of work. So it is coming at a cost, but the cost is a fraction of running a clinical study. So that's maybe important to bear in mind.
Great, thank you, Masood. Let's see, the next question. Do you develop and incorporate full PBPK models for these CYP substrates and inhibitors?
Full
Yes.
PBPK? Oh, sorry.
Sorry, you go on, Jean. Sorry.
No, feel free to chime in. So, so do you mean like minimal versus full PBPK for?
I think that's the implication. Yeah. So I think we do full for all of them, right?
In certain
we
cases,
offer both options.
Yes,
Sorry,
we offer both options. And so depending on the model and depending on the purpose of the model, you would choose minimum or full PBPK. So for example, if we are looking at transporters and that's something you definitely want to go full, for example. It really depends on how you want to use your substrate, probe substrate, or perpetrator.
Great, thanks, Jean. Next one, are there any plans to develop an obese pediatric population?
You go on, Amaka, yeah, and then I can.
Yeah, I think we get this question a lot. So is there work being done on an obese pediatric population? Yes, there is. Is it going to be released in the simulator? Probably sometime in the future when there's more data available, but there's a lot of work going on in the background that is looking at the both the development and the validation of an obese pediatric population. So yes.
Just
here's yeah, go ahead.
Just one comment I just had. I saw somebody spying on my email because today we had a discussion with one of our academic groups, and they wanted to work on obesity in pediatric, and we are going to work together. So yes, it's going to happen.
Excellent, thank you. So we have a lot of questions left, and I'm just trying to cherry pick one that because there's so many. So that's great. We've had such interest. Let's see here. Considering the DDI topic, the hypothesis is to have a test drug that is metabolized by more CYP450 isozymes, and between these isozymes, there is not relevant involvement of one isoform. Oh, I'm having trouble following. So I think in this case, can PBPK model help us understand if a clinical DDI study with a specific, I think what they're saying is that there's multiple CYP enzymes involved. Can we still use the simulator to decide whether or not a DDI study is warranted?
I think the short answer is yes. Yeah, it is possible. And there are several cases that Jean showed. There are more than one enzyme are involved. So you have 3A4, 2D6, 2C9. And another case that she showed, for example, rifampicin, is inducing a number of different enzymes, and we can incorporate them. So, Jean, if you want to expand on that, please.
Yeah, so I mean, just to just to say, like, I highlight the compounds that really target CYP3A4, but that's not the only player in the game here at all. We try to incorporate as much knowledge about each compound into the compound file itself so that you can anticipate how PK would behave. And to do that properly, you do need to account as many mechanisms that are robust and comprehensive. So it's not just 3A4, it's really things that we think could impact variable PK in the population.
One last question here. For pregnancy population, can it simulate cord blood predictions or concentrations?
Yes, yeah, it is possible, and we have extensively published on the pregnancy over the specifically last 2-3 years. There are many models that we have, a range of compounds. We ran very recently a workshop for clinicians that were working in the pregnancy area, and they were interested to see how PBPK can help them to do the dose adjustment for pregnant women.
All right. Well, I thank you, everyone, for a great presentation, panel discussion. I think Suzanne has jumped on so that she can wrap things up for us. But thanks, everyone. Great work.
Awesome. Thanks so much, Mark.
Thank you, Mark.
Can you go to the end, last slide, please? And thank you so much to our esteemed panelists from Simcyp. You guys did an amazing job. Just want to let you know about the next upcoming webinar. It will be on February 7th, where we're going to have a guest from Novartis, Dr. Ioannis Louisis Konstantinidis, and he'll be presenting his a webinar about his work to use PBPK to support the NDA submission of asciminib and inform the drug product label. And if you'd like to come to this webinar, you can register on the Certara website. On behalf of Certara, I'd like to thank everyone for coming and asking so many great questions. Have a great day, and we'll see you next time.